Hello. Our symposium will be about how to use electrophysiology as a tool for global health. Uh, in, uh, in July of 2016, I organized, together with Shekhar Saxena and the leaders of all the major brain projects, a meeting at WHO headquarters. And the big question there was, how can the research that's being done in the big brain projects, the European Brain Project, the US Brain Project, etc., how will that research impact on the global burden of disease? It was obvious that were needs to have translational research. There were links that were missing. Several things have come out of that. One is a special issue of neuroimage, which will come out soon, which is called Neuroimaging and Global Health. And the other was the creation of the Global Brain Consortium. This was in 2019, which in fact has as members many of the leaders of the international brain projects and leaders in neuroinformatics. Now, the basic thing that has to be considered is that this was started by what we call the CCC axis, which is the union of work of Cuba through the Cuban Neuroscience Center, the University of Electronic Science and Technology, where I share time with the Cuban Neuroscience Center, and it's a small photo of my lab, which is an international lab, the Joint Cuba-China Laboratory for Neurotechnology, and also the McGill Center for Integrative Neuroscience at McGill. And this CC axis was the one that then spurred the creation of the GBC. Now, the content of the global presentation, that is all, not only mine, is after this introduction for me to talk about the Global Brain Consortium Open Science Platform. Ming Li from our lab in China will be talking about harmonization of multinational QEEG norms. Then we will be seeing how to try to obtain transferal from EEG to EMEG and back by Usama Rias. The importance of this is that there's some international projects that have electrophysiology, but in the form of MEG, not EEG. Then how we can use, let's say, cutting edge methods, like Riemannian manifold representations of EEG cross-spectral matrices to do mediation analysis with an example from a 50-year-long study of the effects of malnutrition in the first year of life. We will then see how the cutting edge of this work will be when we have a big brain, high-performance computing compatible multimodal pipeline, which Deirel Paz will talk about, and finally I'll give some conclusions. So we can move on to the first part. This is a slide I borrowed from Alan, but captures well what we've the way we think, and it's that if you look at the interaction of genes with the development of the brain and then the outcome and behavior, we're very interested in what are the trajectories that we can estimate, either from longitudinal studies or for cross-sectional studies, where we can have normal development, let's say that's the green line, but then we can have in red deviations from normal development. And of all the tools that we can use to look at this development, there's a series of neuroimaging tools that depend on the cascade of events from neurotransmission to hemodynamic response. But the EEG and the MEG are at a particular location in this cascade that, due to their high temporal resolution and low cost in the case of the EEG, may play a role in having an effect on global health. And that's why we're bringing this topic today. Now, very early on, the Cuban group and New York University, and this was in a 1977 paper on neurometrics, we started using a common idea that in order to measure the deviation of subjects from a normative database, what we needed was to calculate through a descriptive statistic that describes anything, to take out the population mean and divide by the standard deviation, and therefore measure deviation, let's say deviations in terms of probability from a normative database. We later went on and corroborated that age-corrected norms were much better because, and here we can see an example from a paper by Roy John, who was one of the pioneers in this area, where you get much better coverage of normal development if you take into consideration age, and one could add any other covariate of interest. Specifically, we think that one of the most promising approaches is quantitative EEG, which is based on the extraction of descriptive parameters using spectral analysis, and then the construction of normative databases. This is an example with 128 electrodes. This is designed at the Cuban Neuroscience Center, a 128 channel system. Since the late 70s and early 80s, we have a national network based on Cuban technology produced by the Cuban Neuroscience Center. Here we have, for just one of the electrodes, the occipital electrode, the frequency spectrum, and here we have the recording, and we see here the amount of energy or power at this frequency, which is around 10 hertz. This is typical of the alpha rhythm. We can construct, using this value for all electrodes, topographic maps. For any given electrode, we can then construct normative databases, which we will show you a bit further on. So we applied this already in the early 90s to a system of laboratories in the Cuban public health system. We went on to describe what we call high frequency resolution. That is to say, not in broad bands like delta, theta, alpha, but frequency by frequency normative databases. And then we created norms for these databases. Here you see in this graph on the right that you have the log spectrum for O1 derivation for frequencies from 0.1 to 19 hertz, and here from 5 to 97 years of age. This is from a randomized sampling of the Cuban population, in which healthy subjects were selected through a long process to produce these norms. And this is characteristic of the mean value of the log spectrum. We have similar types of curves, which we call EEG developmental surfaces, also for the standard deviation. Therefore, there's for each electrode, for each mean and standard deviation, a developmental surface. Now, we later went on to develop in the middle 80s and methods for doing source localization. This is our particular variety from 1987, variable resolution electrical tomography. Here you see the origin of the alpha rhythm. This was described very early on in the early 90s. And then we applied the same concepts to this type of parameters. Here you see here that we have the norms for, let's say, the O1 derivation. Actually, you can see a movie, which I don't have here now, in which you see how alpha increases in the, let's say, in the occipital areas and then decreases with age. Now, what does that allow us to do? That allows us then, if we have any given inverse solution, to do the same idea of the Z transform, but voxel by voxel, or let's say element of the surface cortex, and subtract mean and standard deviation, and therefore have what we call a Z map. Okay. Now, this concept has been applied. This is an early application in which we were able, using this type of Z maps, to carry out discrimination of subjects that had the presenilin 1 mutation. This is a series of families studied by Dr. Lopera in Colombia, and we did a study where we compared subjects that did not, in a family that did not have the gene, subjects that had this gene, this mutation, and then without disease and with disease, you can see there's an increase in these groups of theta activity. This allows discrimination, though not strongly when you use scalp information. When you use our methodology in the sources, this is like a molecular image using EEG, and here you can see that the discrimination increases considerably, which supports the idea that this technology is useful. We then did a second wave of the Cuban Human Brain Mapping Project. This now was multimodal. We included MRI, blood samples for DNA, the same EEG, with large sample sizes. We then have made this multimodal data set publicly available. This is the publication in Nature Scientific Data, which is the approach we want to take to make our software and our all our software and all our data as public as possible. This is supported by the CCC access I told you about, Cuba, China, and Canada. Here you see the different databases, and our idea is to provide a portal. This is a portal that will allow people to make queries about data sets from the big databases, not only our own, and therefore to then be able to use analytics using high-performance computing from other countries that will allow us then to people with less resources and bandwidth to make this truly global resources. Now, to be more specific, what we have been developing in the CCC is a series of toolboxes. You see there one. We've also, that's the interface for the quantitative EEG, both at the scalp and in sources. And then you can see that also we have been making the data from both waves of the Cuban Human Brain Mapping database available. I'd like to say that we've also developed, as part of this work, a series of toolboxes that will allow us to do the selection of biomarkers. Here are some examples with measurement of area under the ROC curve and so forth robustly. That is to say, using resampling techniques to be sure, and some publications that we've done in this direction. I now turn over the symposium presentations to Ming Li, who will be speaking of harmonization of multinational QEEG norms.
Hello everyone, this is Ming. The topic of the talk today is harmonization of multinational QEEG norms. To understand brain disorders, the normal development of each individual, we can find persons, objects, and help people with mental disease diagnosis. For brain development, many brain disorders are highly heterogeneous. However, the most common statistical designs used in brain disorders research studies and clinical trials, instead of looking at this heterogeneity, they did group average comparison. For example, patient versus control, treatment versus placebo. This comparison hindered the identification of brain disorder and treatment. Now, the increasing available large open neuroimaging datasets provides opportunity to examine patterns of neuroimaging variability in individual patients. Beyond the average comparison between groups, an international norms study is more productive in capturing individual neurobiological fingerprints. Multinational norms are now being a common endeavor. We have effort on the work of brain charts for the human lifespan, which is organized by Richard Best, collected MRI data spanning 115 days post-conception through 100 postnatal years, totaling 122,123 scans from 100,071 individuals in over 100 studies and across 6 continents. We built a standardized neuroimaging-based reference chart to study brain disorders and provided a standardized and interpretable measure of deviates. It may reveal new patterns of neuroimaging differences across neurological and psychiatric disorder emerging during development and aging. Multinational norm study gave a standard brain changes with age, and these changes are associated with functional deterioration and neurodegenerative disease. It is vital that we better understand individual differences in the brain aging process. Hence, techniques for making individualized predictions of brain aging have developed. Now researchers are more inclined to use tools for predicting brain age, and they try to find the aging biomarker that enables a qualification of individual differences in underlying biological aging. And the correlation of neuropathology with chronological age in healthy people plays a crucial role in disease diagnosis. Thus, these papers that analyze individuals who deviate from norms produce more reliable results. In fact, 50 years ago, in 1973, EEG brain age have been studied. Pedro and his colleagues make a further study by using the Cuban Human Brain Mapping Project first wave dataset, and the results produced a high-resolution brain development surface of norms on log spectrum, which described how alpha peak changed with age. Their study indicated that the age effect could not be overlooked when comparing and statistically analyzing. Based on these results, the high-resolution quantitative EEG study eliminates the age influence by subtracting age mean and deviation from log spectrum. The product is called Z-spectrum. Pathology diagnosis can be made based on the deviations from normative aging patterns in EEG. However, this study is limited to one dataset. Building a standardized brain development surface needs a multinational EEG norm study, but here, the collaboration of the multinational project faces a big problem: the existence of the batch effect. What is the batch effect? It can be explained with the story of traveling phantoms in MRI or diffusion MRI study. Datasets collected from different sites on our MRI study show significantly different results, and people make adjustments for the errors produced by external factors by sending either human subjects or physical manufactured phantoms to each site and scan for acquiring the baseline. The batch effect can relate to the equipment's protocols, human errors, etc., and this inevitably can cause reliability problems and hinder the ability for other researchers to reproduce the experiment. Harmonization or standardization of neuroimaging data could counter the batch effect. Thus, comparisons and methods are vital to improving the interpretability of the research findings. To build a more robust standardized EEG norm study, we created a batch effect harmonization work pipeline to counter the batch effect problem, and also enables us to create a global quantitative EEG study to improve the pathology diagnosis. This pipeline integrated data collecting, pre-processing, outliner detection, batch harmonization, pathology diagnosis, and data sharing, 6 modules in total. The multinational team shared multi-site cross-spectrum. The multinational EEG norms datasets from 9 countries, 16 different batches including more than 1,000 subjects for data collection. Instead of collecting raw datasets, we directly collected cross-spectrum and metadata by sending the same calculation code to the collaborations. This code gathers EEG data and stores them in a standardized structure. All datasets are restricted to 19 electrodes and checked for artifacts. The age span for the samples of multinational EEG norms collected ranges from 5 years to 97 years. Instead of working on log spectrum study, we extend our research to cross-spectrum. The sample cross-spectrum matrix is a Hermitian positive defined matrix that does not belong to a Euclidean space, but lives in a Hermitian manifold. In order to satisfy standard vector arithmetic, the cross-spectrum matrices should first be mapped to Euclidean space. Here, for each frequency point, relying on the geometry mean, the cross-spectrum is mapped to a tangent vector space. And here, we first reproduce the development surface which obtained in 1994 in the first wave of Cuba Human Brain Mapping Project and found it concurrent with the one obtained in multinational EEG norms. Do T-distributed stochastic neighbor embedding on Z-score of log spectrum, projecting Z-scores to low-dimensional space, and here we can see there are no batch cluster, which means that log spectrum doesn't have patch effect. Do same procedure for Riemannian vector cross spectrum, the results show that there is batch effect. So, build the standardized norm surface, we constructed our batch effect harmonization model that counters the mean and deviation of batch effect. We also underwent model selection based on information criteria. Our model is more general than generalized additive model for local scale and shape and combat model. Furthermore, our model is more robust and more applicable for other studies. In this study, our model is able to perform outliers detection. This is achieved by calculating the Mahalanobis distance on low-dimensional space Z-score. Here, in the figure which describes the relation of Mahalanobis distance with robust distance, the data points tend to cluster in a straight line that passes through the region. Points that are far removed from this line are generally considered outliers, and these data are corresponding to the faraway cluster. To check the results, first, we can see that before doing the batch effect correction, the Z-score mean value of different batches is not zero, and they distributed on either side of the global Z-score mean value, and the total mean is zero. After the batch effect is corrected, the Z-score mean value of different batches goes to zero. Second, the above conclusion can be verified by looking at the Z-score distribution. Before batch correction, the Z-score mean value of different batches is not zero, and the global Z-score mean value is zero. After batch effect correction, the Z-score mean value for each batch goes to zero. We have proved the existence of batch effect and have used our batch harmonization model to build the standardized EEG norm surface. However, we are not sure about whether batch-corrected standardized EEG norms can actually improve the precision of pathology diagnosis. By computing the Mahalanobis distance of malnutrition Z-scores and norms Z-scores, which come from three different Z-maps, log-spectrum Z-map, Riemannian cross-spectrum Z-map, and batch-effect-corrected Riemannian cross-spectrum Z-map, we see that firstly, Riemannian cross-spectrum performs better than log spectrum. Secondly, the batch effect harmonization shows the best results for improving the pathology diagnosis. From multinational dataset collection to pathology diagnosis, the normative harmonization of multinational EEG norms will not only solve the problem of data pooling, but also will enable us to build a standardized EEG brain chart in order to better track the performance of each batch. Thus, it accomplishes the privacy-protected dataset sharing work and improves the accuracy of disease diagnosis. Further, we want to extend the multinational EEG norms by including more samples and multimode datasets to complete the multinational EEG norms model. And here, I want to mention that we will not bother by the time-consuming problem and the high-dimensional problem because we have the technique support of ultra-fast, non-parametric regression algorithm which created by Yin Wang, and this allows us to handle tremendous datasets in absolutely short time.
So the first thing that we need to know about is, are the sources of EEG and MEG rhythmic activity are same or not? So for that, the basis of our study is that since both phenomena are capturing the same cortical activities or brain activity, so the sources generated from both of them should be same. However, there are many circumstances that change that thing. The volume conduction effect, the both modalities phase is different. The measurement noise might be different because of the engineering of sensors and physics behind capturing the signals, and the filters of both sensors have different transfer function and amplitude response. So what we did was we compared two source spectra using the pipeline of big brain HPC compatible multi-model pipeline that we have developed at. So now that we know that both source spectra are different, what we did was we applied some regression models and to reduce those differences or to regress the EEG source spectra and MEG source spectra and try to visualize those as frequency spectra plots. So what we did was, before comparing, we conducted two different regression models. One was spatial regression model, and here's how this model looks like. Here you can see that we are regressing EEG or MEG, and once we have the regression coefficient, we applied those coefficients to the MEG spectra and try to reduce the differences. And the second regression model that we used was Xi-based frequency model, and that frequency model is basically represented here in this equation. Here we are using a Xi process that explains the background activity of EEG and MEG, and using that Xi process we regress EEG or MEG and have our regression coefficients, and using those regression coefficients we try to transfer from EEG to MEG and see and plot them in form of frequency spectra. So here is the frequency spectra before the corrections. You can see that for all three solutions, E Loretta, LCMV and BCA Loretta, the source spectra were different. They had different slopes, but when the regression. So what we did was we compared both source spectra using statistical tests, and we carried out some statistical comparison, and based on those statistical comparison we found the same trend that we observed in the visualization of source spectra. We conducted a t-test. This is how conventional t-tests look, but what we did was we first permuted both source spectra. We combined them and do a permutation, and then get two equal chunks out of that permuted EEG and MEG source spectra. Then we applied a t-test on those two chunks, and then later we conducted an inverse probability and distribution process on the threshold level 0.01, and to achieve, and from that we achieved the threshold threshold TH alpha. And when we applied that threshold on the t-test, the t values we have, we found that we labeled that the values that are greater than the threshold will be significant, and otherwise they will not be significant. Then we plotted those source spectra on the cortex, and this is how that looked like. You can see that both modalities are significantly different in almost all brain areas. And when we did the corrections and plotted them again so once we have established that we can transfer, or we can use, reduce the differences between EEG and MEG source spectra, we try to find or propose a transfer function that will help us transfer from EEG to MEG. And for that, we first decompose both spectra using this model. Here on the left side, we have the source spectra. On the right side are basically the components that we are trying to estimate. So the first component we estimated was Xi process, and that's the background activity, and we estimated that using the spline fitting. The second component we estimated was basically the individual differences of the subjects of EEG and MEG, and that was simply computed by taking a mean at all subject level for both spectra. The third thing that we estimated is basically common topography. Common topography basically tells that we are capturing the same cortical activity in two different conditions, which are either captured by EEG or captured by MEG, and the scale value will, when we apply that scale value on that common topography, it will get transformed the common topography to EEG topography or MEG topography. We computed that using covariance-free stepwise common principal component, which give the eigenvectors and eigenvalues for the variables captured at different conditions, the same variables captured at different conditions. So what we did was we first made some initial estimations for all three components and then iterated them continuously until we converge. So we subtracted the initial estimated values from the original spectra, two of them, and then estimated the third, and we do that continuously until we converge. And the convergence criteria was basically a relative error from previous values. This is how we then this is the Xi estimation. You can see the estimation of EEG in red and blue for MEG. This is the individual differences. This is the Xi process. This is the common principal common topography with the scale value smaller for MEG, larger. So once we have estimated the individual components that will eventually reconstruct the source spectra, what we did was, for example, for MEG, we subtracted those components from MEG source spectra and added the components of EEG source spectra to that, and that will transform the MEG source spectra to EEG source spectra, and same is true for EEG. So this is how the source spectra for MEG look like. The blue one is the original source spectra and the red one is the one that we have created using the EEG source spectra and applying the MEG source spectra values to that. And same is the case for EEG. The blue one is the original source spectra and the red one is the one that we have created from MEG and applying the EEG values to that. So in conclusion, we can say that yes, both spectra are different. However, these differences can be removed, which was evident in regression models, and the decomposition and reconstruction of EEG from MEG source spectra or vice versa is possible, which we have seen. So what we are right now, what we are doing is we are trying to apply this model to a bigger data set, like for 1,000 subject or somewhat 700, 800 subject, to see that if it holds true for them. And those subjects we are trying to gather from different EEG datasets internationally, so that our model can be validated.
Our study of high-dimensional and Riemannian mediation is based on the study of malnutrition in Barbados. In this study, subjects suffer from malnutrition during the first year of life, and this is the cause in our mediation diagram. As the effect, you have the cognition, and as the mediating variable, you have the brain state that is captured by neuroimaging variables. And the theory is that malnutrition causes changes in the structural and functional aspects of the brain, and that cause cognitive changes. We have done point estimates as well as longitudinal analysis for testing this hypothesis to understand the causal mechanism behind protein energy malnutrition. Our latent mediator and cognition variables were measured by EEG soft neurological science and GTE, and by intelligent test subscores. We have screened these measured variables for no causations, resulting in 5 out of 10 variables for GTE, 7 out of 16 from soft neurological science, and 145 out of 912 from EEG at the significance level of 0.01. We did this using the R-package global test, and also we run 9 different global tests and discarded any variable that were not significant at alpha 0.01 in any of the 9 regressions. We evaluated 4 models of mediation, the first 2 being corresponding to brain state 1978 and cognition 78, as well as brain state 2018 and cognition 2018, and the other two corresponding to the combinations of these variables. In this case, 1978 and cognition 2018, and in the last case, cognition 2018, but with both brain states from 2018 and 1978. To assess the robustness of these models, we employed sensitivity analysis. This sensitivity analysis can be measured with the parameter ρ. That is nothing more the correlation between the regressions between exposure, mediator, and mediator outcome. And a not-zero value of ρ can be due to some unmeasured but common confounding factor between mediator and outcome. If the average causal mediator effect can hold its direction to the reasonably significant value of the sensitivity parameter, that means that a robust and stable estimate. These are the results for the first two models, when we have brain state and cognition from 1978 and brain state and cognition from 2018. In the middle column, we can see that we have average causal mediator effect, while the average direct effect is almost zero in both cases. And in the last column, we can see that using the sensitivity parameter ρ and the average mediator effect, we can reject the possible inclusion of unmeasured and confounding variables. And we can arrive to the conclusion that malnutrition causes changes in structural and functional aspects of the brain, and that in return cause changes in cognition. For the last two models, we evaluated the brain state from 1978 and cognition 2018. And in the last model, we evaluated the combination of brain state 2018 and brain state 1978 with cognition 2018. As you can see in this graph, there is a very strong average causal mediation effect, while in this case there is no mediator effect, while all the effect is through the direct effect. And we can also look at the sensitivity analysis to reject the possible inclusion of confounding variables, as can be seen in this graph. And we can arrive to the conclusion that changes in brain state caused by early childhood malnutrition are lifelong and not completely reversible without any interventions. In order to do the Riemannian manifold mediation, we begin with the time domain EEG signals. After applying the Fourier transform, we are in conditions to obtain the EEG cross-spectral matrix that form a Hermitian positive definite Riemannian object. In this object, you have in the first two dimensions the electrodes, and in the third dimension, you have all the frequencies. This object will be the mediating variable in our mediation analysis. We developed a pipeline to map from the Riemannian manifold to the Euclidean space. This mapping is possible using the mean of the manifold as well as the logarithmic matrix operation to map from the manifold space to the Euclidean space. Once these points are obtained, we can follow up to the vectorization. The vectorization is nothing more than taking the upper triangular of the matrices and using the mean of the manifold, obtain a vector for each of the cross-spectral matrices. So in the end, we begin with a 3D object corresponding to the cross-spectral matrices and end up with the 2D object after the vectorization. After applying our method to the study of the effect of malnutrition in the brain, we were able to find that both activation and coherence of the EEG spectrum mediate the effect on school-age cognition of malnutrition during the first year of life. Can be seen in the distribution and significance of the mediation effect seen on the combination of the 10 of the combination of electrodes conforming the 10-20 system.
Hello everyone, on behalf of our team I will present today our work developing standard neuroinformatic facility for the registration of histological and in vivo imaging of the brain, a work coordinated by Professor Pedro Valdes Sosa from the Cuba China Lab for Frontier Research in Translational Neurotechnology. The Human Connectome Project, ACP, have introduced such requirements before, as well as specific standards for data acquisition that are hardly followed. International initiatives have adopted this style, such as the Healthy Brain Networks, the Global Brain Consortium, and the Big Brain Highball projects. We are appointed by these and to fulfill robustness and replicability, somewhat challenging for data acquired with diverse standards, equipment, formats, and protocols. This is coined legacy data. We have introduced ACP-compatible modeling of the MEG and EEG sources, head and lead fields for legacy database. Following: left branch 1. Structural MRI processing that produces the ACP structural spaces and features in cases when T1 and T2 are available, and also when only the T1 is available. Also using template MRI when the individual subject image is not acquired. 2. Right branch, processing of sources, head and lead fields using outputs from a structural MRI that are registered to the legacy MEG and EEG geometric files, also using standard layouts when no geometric file exists. This is made replicable and robust under many conditions using a diverse repository from the neuroinformatic ecosystem. We introduce a quality control loop incorporating quantitative measures and qualitative indicators for detection of artifacts in the ACP MEG outputs. An intensive try and catch loop performed for large databases helps us to incorporate the diversity of functions for corrections or to find alternative processing paths. Some indicators to be controlled are: 1. Cortical registration and cementation. The structures with artifacts are rejected or substituted with a template. 2. Sensor layouts that are correct and then used to recompute the lead fields. 3. Lead fields that are rejected or recomputed after automatic correction of the head module. We have prepared a release of the Healthy Brain Network dataset with our pipeline, which includes legacy MRI and EEG, as well as other type of images. Here we show the processing workflow adopted for the processing of other databases too. In the quality control loop, we have rejected a number of cases to release only quality ensured structural outputs. In the future, we shall incorporate ACP-compatible processing for legacy fMRI diffusion weighted imaging data, such as those of the Healthy Brain Network database. We introduced the BC Vareta pipeline that includes novel methods for MEG and EEG source analysis of activity and connectivity. BC Vareta mathematical sparsity priors control directly the distortions of source spectral variances and covariances, unlike classical methods. We have introduced surface-based structured sparsity priors into BC Vareta, which reduce the bias of sparsity and provide cortical anatomical information, such as cortex surface-based neighbors, depth compensation by curvature, rotational invariance for fields, and parcellation smoothing. It's been our task to study the behavior for BC Vareta in large dataset of realistic simulations. We have adopted a concept from a previous study. This is to create individual source, head, and leaf field models and a 19-channel low-resolution pseudo-EEG. This pseudo-EEG is, in this case, expected EEG for the ACP-MEG data, which is obtained from the MEG sources that are estimated with an independent method. We then compare BC Vareta in the ACP, MEG, and pseudo-EEG data and obtain the optimal configurations, the parameters, and their range based on the values of quality measures. We have produced the registration of the big brain onto the ACP space, which we have based upon a hybrid image of the big brain and the ICBM MRI template that are weighted in T1 contrast. With this registration, it is then possible to obtain the concurrent mapping of any structural and functional feature onto the individual ACP and big brain spaces. We show an example of MEG-EEG source imaging that is practiced onto the individual and big brain spaces using ACP-MEG and BC Vareta. Source imaging from both is then mapped across all spaces. There is no other software that carries out this functionality. A strategy has been projected to inform subject-specific neural models. This will reveal the true space of parameters describing brain states. The steps for this are: A. Define a neural model with cortical layers and columnar connectivity that are obtained from big brain. And B. For instance, this can be the ultra-large Janssen and Reed neuron mass model that was developed by our team. The model is informed by C. Source, head, and leaf field obtained from ACP-MEG, and then D. BC Vareta source imaging and connectivity. Discovery of the parameters shall be done by 1. Expanding in spectral mode the neural equations, and 2. Inferring them from the BC Vareta results. Then E. The BC Vareta results or neural parameters are projected across all spaces, individual, ACP, and big brain.
To conclude this symposium, which has covered aspects from basic methodology to creation of norms and the use of sophisticated methods to do inference using the EEG, I would like to say and remind you again that the Global Brain Consortium is opening collaboration with many other groups, and our basic idea is to achieve a final goal, which is to democratize the use of quantitative EEG methods in electrophysiology, together with other multimodal techniques. Our basic concern can be summarized by saying that, as we discussed in the WHO meeting, there is a divide between neuroscience and having an impact on the global burden of disease. And the way neuroscience basic research achieves an impact on medicine is through neurotechnology, that is to say, new methods for diagnosis and treatment, which is known as translational research in neurotechnology. But unfortunately, it's limited to the third or maybe secondary level of medicine. It is not until we create tools and engage with the primary health level that we will be able to have a truly big impact on the global burden of disease. I want to give an example of why this is important. Consider now the worldwide raging epidemic of COVID, a problem that will affect millions of human beings is long COVID and what I like to call COVID-induced brain dysfunction. And there's hypotheses that the virus can enter through the olfactory nerves, the brain directly. Other people think that it's blocked at the blood-brain barrier, but the immunological aspects then, through the inflammation of the choroid plexus, produce problems. There's the cytokine storm. There is problems with stroke and other cerebrovascular accidents. There's, in the acute phase, many symptoms: hyposmia, anosmia, hypogeusia, ageusia, headache, dizziness, brain fog, cerebral vascular disease, encephalitis, myalgia, even Guillain-Barré syndrome. Now, in long COVID, how many of these persist? There's intriguing work done in electronic records that give alarmingly high rates of posterior increase after COVID of some neuropsychiatric diseases. However, there is a problem, which is how do we measure in so many people? What is the tool that we would have accessible to be able to separate social, psychological, and actual damage to the brain? So Cuba, in its national program for the neurosciences and neurotechnology, has a project. It's being directed by Dr. Roberto Rodriguez from the Cuban Neuroscience Center. And the idea is to carry out a longitudinal study of long COVID, of COVID-induced brain dysfunctions that will have standardized, that has standardized tablet-based neuropsychological battery, that it has schedules for clinical assessment, psychiatric assessment with the SCAN method. It has objective quantitative electroencephalography, and we are doing neuroimaging in a part of the subjects. Now, the important thing here is the following. We are studying now for 6 months, and it will be for several years probably. This says 1 year. It seems it's going to be necessary for more time. At least we studied with 400 convalescent subjects and controls. Now, the controls are matched in sex, age, and education, and the controls are people who had, let's say, were in contact but had a negative PCR, so they went through the stress that the other people went. The details will be coming soon. We're working on a Latin American project of this sort, but we did find excess delta activity with respect to controls. And okay, when we see how sensitive and specific this is a possible example of an additional tool that might help, besides questionnaires or whatever, to decide exactly what flavor of long COVID people have. This is from the source analysis. You can see that there's those straight lines are the significance levels overall, the brain of voxels, and then you see the excess slow wave activity and beta activity. So this is the end of the symposium, and we hope you've been interested in this, and we're open to collaborations on any of the topics that we have mentioned. Thank you for your attention.